Following is a conversation with Dr. Alan Walker, a senior lecturer at the Rowett Institute for Nutrition and Health at the University of Aberdeen. Dr. Walker has been working in the microbiome field for the last 20 years, and over the course of this episode, he shared with us some of his insights into how the field has developed, how new technologies have improved our understanding of the microbiome, and we also spent a lot of time having some fun debates about pros and cons, some of the benefits associated with microbiome modulating therapy, and also where some of the myths and misconceptions lie. So for the generalist who wants to learn more about the microbiome, I think this is an excellent episode. We also get pretty deep and pretty technical in some areas, like, for example, microbiome sequencing, next generation probiotics. Can the microbiome cause obesity or be contributing to obesity? There's tons in here. And we go right the way across the microbiome field, talking about diagnostics, at-home microbiome testing, the use of faecal microbiota transplantation. What is a healthy microbiome? So absolutely fantastic, lots in here. And because Alan's been a friend and a collaborator for, maybe 7, possibly longer now, years now, we've got a really nice dynamic over the course of this conversation. We push each other. We have strong debates. It's not in any way, shape or form confrontational. It's two friends having a really fun, in-depth discussion about the microbiome. So I hope you enjoy it. I'm really appreciating all the feedback you've been giving the podcast as well. As I came into the podcast studio today, apparently one of the listeners had been asking if they could be connected to me. So thank you so much. Please just reach out on socials. This will be the last podcast that I've recorded in 2023, so I wanted to take this opportunity to thank all of the listeners and supporters of the podcast for everything you've done to help us build the name, the brand, and to get the message out there around microbiome being critically important and gut health being really important for wider body health. Please reach out on socials or at james@insidematters.health if you'd like to connect and have any discussions about the microbiome. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. So when I first met you, I was a young man.
Still are.
Although it's feeling less and less like that, I have to say. There's some grey hairs coming.
That's how life goes. Yeah.
And I'm now strategically using a photo from a year and a half ago in my presentation conclusion. So there you go. That just tells you things are...
Well, academics use photos from 30 years ago, so you're...
but back then, the microbiome was a thing that people were aware of. Mm-hmm. But there certainly wasn't the same kind of interest in gut health and so on. You feel the same?
Yeah, I think probably over a slightly longer time frame, I guess. So, because I've been working in this area since I think about 2002, so just over 20 years now. So yeah, if you don't feel like a young man, I definitely don't. When I first started, it really was kind of like a cottage industry for science. There was maybe one paper a month came out, and that would be on any gut microbiomes, cows or chickens. But there's a paper out this month. Last year I checked, it's over 1,000 every month.
Wow.
So it's really sort of snowballed massively over that 20-year period. I like to think I'm personally responsible for it, but sadly no. It's been there has been this sort of constant exponential growth in the field, ever since sort of the mid 2000s, really.
And how do you keep up to date as someone who's immersed in this? Because you can't read 1,000 papers a month.
No, you can't. So the honest answer is no one can keep up to date. So you absorb knowledge through basic reading, listening to people, going to talks, going to lectures. But you'll always miss stuff, unfortunately, in the field. But so it's a very different, like I say, area than when I started. Before, you kind of felt that you could absorb everything and you learned everything in the field. Nowadays, you pick out the landmark papers and you find stuff that's interesting. But no, it's a lot more difficult to keep up now.
Can you try and summarize what the field has learned over the last 20 years in sort of big picture terms?
You know what I find most interesting is that actually, if you think big picture terms, we knew quite a lot of this stuff already 20, 30, 40 years ago. So if you read sort of reviews from the 1970s about the gut microbiota, there's lots of stuff in there which is still 100% accurate, about the importance of short-chain fatty acids for health, the breaking down of fibre. A lot of this stuff is already known. I think one of the myths in the field is that, that microbiome is a new field. And it really isn't. It goes back over 100 years. E. coli was first discovered in the late 1800s. bifidobacteria, these really beneficial bacteria from babies, also discovered in the late 1800s. So, the field has quite a long, rich history going back. It's just, as I say, it's just really exponentially grown over the last 20 or so. And I think people have lost sight a little bit, actually.
Wow.
There was a lot of received knowledge already before the field took off. And I think sometimes we do a disservice to all that really brilliant research that was done, all the way back to Pasteur and all the sort of people back then in the 1800s.
So maybe we can kind of summarize it with the question, why is the microbiome important?
Yeah, no, it's a question that we're all asking in the field, I guess. I think there's multiple answers to this question, but so I'll try and hit it by layers. I think the thing I try and get across to students and people who I talk to in public engagement is this is a microbial planet, right? Microbes were here billions of years before we were, right? And so anything that's evolved into this world has had to do so in the presence of microbes, okay? And so, we as human beings did not appear in the world in a vacuum. We have to deal with these microbes. They were here first. It's their planet. And so as our biology has evolved, our bodies have evolved over time, we've had to evolve to deal with these microbes. Over time, we have selected ones which aren't going to kill us off, and they've selected us. If you're a microbe, if you kill off your host, you've got to go and find a new one. It makes sense to have a host that you can live in relative sort of peace and harmony with. It's a stable place to live. It's a nice environment for bacteria. It always kind of blows my mind that across the entire world, places a bacterium can choose to live. Up your backside is one of the favourite places they have. You have some of the highest density microbial communities are inside our bodies.
Right.
And the reason for that is
in
it's
the
a
whole
warm
planet. In the whole planet.
Anywhere, right?
In the universe.
In the universe, who knows? I've not sampled in the universe, but if you think about it logically, it's from a bacterial point of view, it's a very nice place to live. It's a stable environment. The temperature doesn't change. It's not like being out in the soil somewhere when you go through the frosts. You get a relatively constant supply of food and nutrients.
Right.
So from a bacterial point of view, actually, it's an amazing place to set up home. Just it's poo. So from our point of view, it seems weird. But from a bacterial point of view, it is actually a remarkably stable and welcoming place to be if you're that kind of a bug. So So as I said, to go back to your question, how important for health is, they're fundamental to our physiology, the way we are as human beings, right? So I don't think it's a surprise then that these creatures that we share a world with, have played an important part in the way we function as human beings. So, that can range from protecting us from pathogens, and we know that if you give, say, mice antibiotics to wipe out their microbiota, they become 100,000-fold more susceptible to infectious disease. You can knock out bits of their immune systems and you don't have that level of, reduction in protection. Wow. So they're hugely important for protecting us from some of the nasty germs. They help us break down fibres and things that, otherwise we would poop straight out because we don't have the enzymes to break those down. They they teach our immune system, they keep us primed, and a whole host of other things, which we're only just learning, the more and more research is done. So fundamentally important for our health. I have to say there's a flip side to that. So germ-free mice, the mice that you grow in a lab, they live longer, not shorter lives.
They live longer?
They do live longer.
I thought they lived shorter.
No, they live longer, yeah.
But how do they live? Do they live better?
So they're in a bubble, so they're never exposed to microbes. And so their immune system is underdeveloped, so they don't have to waste so much of their energy, having an immune system, for example. That would suggest that overall the net effect is that, microbes actually are not the beneficial thing we all talk about. But that is in a little bubble which does not reflect reality in the world. Like I say, that in that little bubble these mice grow in is not like the world. We're on a microbial planet. They are not. So the world we're in, the microbes are hugely important for us.
I had always thought that these germ-free animals lived less long.
No, they live longer. Yeah, that goes back decades. That finding was first discovered.
But if you move them from their germ-free environment into...
Oh yeah, then they're in deep trouble.
Then they're done.
No, they're really because their immune system hasn't developed to deal with this microbial world, like I say, that we all live in. They have underdeveloped immunology, and so they really don't know how to deal with microbes when they hit them. So like I say, within that bubble, that would suggest that overall, if you lived in a magical world without microbes in it, then we would live longer, happier lives potentially. But the world we're in, that's not that world, right? We don't live in that bubble. So
just
for
a
us,
couple of thought experiments, maybe. In
sure.
that vein. I mean, this is really hypothetical now.
Yeah.
But can these mice actually digest and break down, the fibers?
So the answer is no, they don't. They have to consume more. to get the same amount of energy because they don't have the microbes to break down the fibre. They actually have to supplement their diet with the B and K vitamins that their microbes would normally provide them with. So no, they are missing things because obviously those mice didn't evolve in that bubble either. They evolved in a microbial world too. So actually what you're putting them in is a very unnatural environment for them. So no.
And to bring it back to what you said at the very start, this is a microbial world.
It is, yeah.
So translationally, it's not really that relevant an experiment, I suppose.
Exactly,
but it's an interesting...
It's just an interesting observation. We talk about microbes being so good for us, but actually, if we lived in a world without them, we might actually do better.
So how does the microbiome develop then over time?
Yeah. So I mean, for me, the most interesting thing about the microbiome is the complete randomness of it. Right. So every single person on this planet has a different microbiome, right? Think of it as your own smelly fingerprint, right? And so we know a little bit how that comes together. Some of it is down to host genetics, but actually a very small proportion, we're talking less than 10% is explained by your genes. Some of it is what we eat, and in babies, what we eat is super important to that microbiome developing, and I'll talk about that in a second. But really a lot of it seems to be just what you're exposed to. Right. So when a baby is first born, it comes out. If it's born through the vaginal tract, that's the first microbes it's exposed to. Some of the bugs from there will get in. Anyone who's been at a birth will know it's quite a messy process.
I've delivered a baby.
There's poo there. It's messy. Yeah. So those bugs are the first bugs that get into the baby, right? If they're C-section, slightly different. Ones that come from the skin, come from the surgeons operating, slightly different. However, within a very, very short period of time, talking days, those are replaced. And that really is driven by what the baby eats. So if the baby is breastfed, get lots of these bifidobacteria that people may have heard of. They're in sort of probiotic-y type drinks. And generally are thought of as quite benign, quite helpful bacteria, I guess, and may actually help the baby's immune system to develop and train them on a slightly less aggressive set of bacteria. If you're not breastfed, if you're formula fed, you have some bifidobacteria still, but other types of bacteria also tend to come in as well. A bit more of a diverse community. And then after weaning, you get this big expansion in the diversity of the microbiota as we start consuming different fibres and providing different energy sources for the microbes that live there. So that process goes on for the first few years of life. And then once you get to sort of adolescence, adulthood, it's relatively stable over time. So we can go back to donors we've had for years and years and years, and I know which donor is which by their microbiota profile. I mean,
really?
It fluctuates a little, whether you had Weetabix for breakfast or yogurt, whatever. But overall, the types of bugs that are in there are relatively stable over decades and decades. And that process of how you end up with that stable community is still not fully understood. And a lot of it does seem to be just what you're exposed to. Now, you can have identical twins in the same house, same diet, exposed to the same bugs, and they still end up with a unique microbiota. Okay, it's more similar than two random people that have never met before, so there is some sharing. But it's a fascinating process we don't fully understand, but in the end, everyone ends up with this sort of unique configuration of microbes, which is interesting, but makes this research field so difficult to study, and we'll come into that, I guess, later on maybe.
What impacts the microbiome, in a big way, and do people always revert back to what you're describing as that stable? You call it a core microbiome? Is that what the field use?
Yeah, I guess core. Or I guess that's probably a fair word for it. Yeah. So the interesting thing is that you can really perturb the microbiota. You take antibiotics, for example. But the really interesting thing is, unless you're repeatedly hit with antibiotics over and over and over again, like someone who's in a hospital environment, your microbiota largely will grow back over a few weeks. Now, not entirely, but mostly it bounces back. It's quite resilient. And to me, that's the most interesting thing, right? There's obviously some left over behind. They've set up home with your immune system recognize these bugs, and you tolerate each other, and it goes back to that sort of steady state, Again, you change your diet, you can have some effect on your microbiota, but if you go back to your old diet, your microbiota will snap right back as well. So, when we talk about changing your microbiota by diet, I think there's a lot of easy sells in the field, here, try this wonder diet.
Right.
But, great, but as soon as you go back to your old diet, so if you want to change your microbiota for life, you probably have to change your diet for life, which is a much harder sell than, do this for a couple of weeks and you'll be fine.
Or take a product that contains a full spectrum of microorganisms once every few months.
Yeah.
Not saying that's what we should do.
No, but will they colonize, yes or no? And, a lot of those bugs won't colonize. As soon as you stop taking the product, they're gone. And so I think this has been an issue in the probiotic field for a long time is, as soon as you stop taking the probiotic, it's gone. I told you that your microbiota protects you from invading pathogens. It also stops potential nice guys from colonizing too, because they've got their house, it's their place, get out. And so, some will stick around. You do pick up bugs as you age and go through life. You pick up from the environment here and there, but it's a much slower process than when you were really young. So it's tough. It's really tough to predictably and reproducibly alter a microbiome. And I guess maybe we'll come back to it later. But yeah,
it brings me to a question that hopefully an expert like you should find very easy to answer, which is, what is a healthy microbiome?
This is a good question. I get asked this quite a lot. What is a healthy microbiome? And given that everyone has a different microbiome, everyone in this room here, you, me, we all have different microbes. The answer I always give is one that doesn't make you sick.
Interesting.
You ask me to define what is a healthy microbiome, what does it look like? There's such a spectrum in people and what their microbiome looks like, especially down at the species and strain level. It's if it's making you happy, healthy, you're not sick, then for you that's probably a healthy microbiome.
Beyond that, though, could you go a bit deeper in terms of constituents, functions, presence, absence?
No. That's the thing. I think people will tell you they can. I don't believe you can. I think, there are certain key functions like short-chain fatty acid production, whatever, that these microbes, fine, everyone knows these things are good for you. But, lots of different microbes make short-chain fatty acids. I would argue that, having a decent amount of butyrate producers, butyrate being one of the short-chain fatty acids, is probably a good thing on balance.
Sure.
Other people would argue that butyrate can be bad for you, because it has sort of effects on host cells. So generally, we're having a debate here where there isn't a consensus, and there isn't a consensus for a reason, right? Which is that the microbiome is so complex and has varied so much between people that actually it's been very, very difficult to say, look, if that person has this microbiome configuration, they're definitely not well. If this person has this, they're definitely fine. And it's just been very, very difficult to get to that stage.
But we've identified across a large number of what I'll call kind of case control studies that there are distinct differences between people who have disease and people who are quote unquote healthy.
So I'm going to put a question back to you. What do you mean by distinct?
Distinct. So what I observe, what I've seen a lot of, is most notably and most consistently reductions in diversity, defined by the ecological indices, Shannon, inverse Simpson, all the things that you.
At what point does that become diagnostic?
Well, we'll get to that. We'll get to that. And I'm being interviewed now. Testing me. I see very frequently differences in relative abundance in particular genuses. So, for example, proteobacteria.
So I agree. That's the one thing I would say. Diversity. That fluctuates. We have a lot of studies, for example, where we feed dietary fibre to people, and diversity goes down, not up. Right? And
why? How?
Well, I'll tell you why. So if you say you fed resistant starch, okay, I think you talked about this with Harry Flint when he was on. Ruminococcus bromei is a great resistant starch degrader, right? So if you have Ruminococcus bromei in your gut, that will likely respond to eating lots of starch, right? So it will go up.
Okay, I got
and so it becomes 25% of your microbiota. So everything else drops. So your diversity score goes down because this one bug has taken over, right? So diversity being high always equals good. It's one of these. I guess what if you talk to me, the one thing that hopefully comes across is that in the microbiota space, simple statements are very rarely always true. You always need that nuance with microbiota. So usually, yes, a higher diversity is associated with health, but in and of itself, it doesn't mean you're definitely healthy.
I got you. No, I fully agree with that.
But the proteobacteria point is a good point. I think, proteobacteria, Enterobacteriaceae type opportunistic pathogen type bugs, them sneaking up in abundance is probably not a good thing, right? But there are people who are ostensibly healthy walking around who have, say, 5-10%, and they seem to be all right. Now, again, it's not at a point where if you're trying to talk about the microbiome as a clinical feature, where you can go, right, you've got this profile, you've definitely got disease X, we need to treat you with treatment Y. And I don't think you will ever get that level of precision for any disease in microbiome, I'll be honest.
Let's debate that. I'll just share some more insights because I think I have a bit of an unfair advantage here insofar as we profile, I think, more healthy stool samples than maybe anyone else, potentially in the world now, actually, with open biome ceasing to exist, which is I think is pretty cool. And without delving into anything that's, commercially confidential or strictly proprietary or a trade secret, we do see some fascinating trends, and consistent trends.
I'm going to call you on trend.
Okay. Trend. Okay. we see some consistent findings across all the people who filter through our process, which arrives at a cohort who are healthy. So without giving too much away, diversity is consistently higher on these indices than people we've sampled elsewhere.
Yep.
The proteobacteria thing is consistent insofar as we see less than 5% relative abundance pretty much every single time.
Yep.
The fascinating thing, though, is we do see transient carriage of certain things. They appear and they disappear. And again, I can't go into too much detail, but I think in people who are unhealthy, this transient carriage of pathogens or pathobients, can you call them that? Is that a word that we like?
I call them opportunistic pathogens, but that's fine.
In someone who's maybe not healthy, who maybe has reduced diversity, they can somehow find a way to kind of stick around.
So I think, like I say, I'm not here to be a contrarian. I really don't like contrarians, honestly. It's contrarianism has gone crazy over the last couple of decades. People just say things just for the sake of it. I think what you're saying there's definitely merit in it, right? Absolutely. It's well established now across a range of disease conditions. Diversity tends to be down. Enterobacteriaceae tends to go up. Now, I would ask the question: is that a cause or a result of the disease? And I don't think
that's a tough one.
There, if you have inflammation in your body, inflammation will dampen down the growth of many of the beneficial bugs in your gut. They're more sensitive to it than things like Enterobacteriaceae. And it may just be you've wiped everything else in a little bit, and Enterobacteriaceae takes its chance. Now, I do think there is potentially you get this sort of negative feedback loop because they are a little more pro-inflammatory, these Enterobacteriaceae. So I can see how you get in a situation whereby these do a little better because of inflammation. They trigger more inflammation, which means that good bugs never come back to the same level. And so I do think things like that are probably having a role to play in things like inflammatory bowel diseases. I'm not going to argue that point whatsoever. Whether you can take that situation as a diagnostic and say this person X definitely has disease Y, that's what, with the level of precision required for clinical practice, that's where
that remains to be determined. I'll just say one more thing. I think somebody somewhere in the world, and if you're listening and you've got the ability to do this, please do it. I hope we'll show somehow through one of these prospective longitudinal studies that you do get a proteobacteria bloom before the
onset.
That would be so cool. Now,
I agree.
Why it's bloomed, we don't know. But that would be awesome.
I mean, honestly, I agree. I think absolutely. I think longitudinal studies, given the variation in individuals, following people long term, so they almost act like their own control. That is the way forward for the field. Because if you take a group of people X, group of people Y, and just compare them, you will find differences in the microbiome 100%. Just by random chance. It's these longitudinal studies that you're talking about, where you actually get a sort of preceding event that will hopefully lead you to something that's a bit more concrete. So, and the field is doing that. To be fair, I think for 10 years, a lot of people just sampled a bunch of people and went, look, I did this, everyone did it, right? It's what was done. The technology was new. It was the first thing to do was just, look, can we see disease X, Y, Z? Do they have different microbiome? But actually that correlation is not necessarily meaningful. And I think the field as a whole needs to move from correlations into mechanisms and actual consistent patterns that, come across the whole repeated study sets.
At the start, we talked about the kind of 2 decades you've had in the field and what's changed over time and what we've learned. That seems to be quite an interesting trend. It might be worth just unpacking a little bit, the correlation thing moving to causation. What does that mean? So
I mean, for me, the biggest shift was the technology-driven change, So the field, I'm actually really fortunate in this respect. So I was working at the Wellcome Sanger Institute, which is down in Cambridge. And that is a sequencing center. It does lots of DNA sequencing. And so I just happened to be there doing my postdoc work at the time when all these new next generation sequencing technologies were invented.
Just one point of clarification. Was that originally invented for human DNA?
For any kind of DNA sequencing. So so obviously DNA is in everything, everyone. And so anyone who's interested in the biology of an organism can sequence their DNA and learn more. So these new techniques, basically, I mean, I cannot get across, I guess to a non-scientist who might be listening, how exciting that was. So, I mean, I'll try and put it into context for you, right? So obviously, like I say, when I did my PhD, at that time, maybe there was like a few dozen bacterial genomes in the world existed, a very small number, right? And they took probably around about £500,000 each, just under maybe, and years to do, right? It was like, one genome was a massive, big deal, right? And I think at that time when I finished my PhD, there was maybe 2 or 3, 4 gut bugs, E. coli, Bacteroides fragilis, and a bifid had their genome sequenced. And that was it. right?
That was the entirety of the collective global knowledge.
Yeah. And then within 2 years, the cost went from about £500,000 in years to do it to £40 in days.
Wow.
Right? And so I cannot explain to you how much that changes the rules of biology and what you can do as a scientist. Experiments that were literally impossible 3 years ago are now just routine and easy. And so the excitement of being in that institute at that time, a sequencing center set up to sequence DNA when this revolution happened, journalism as a privilege, absolute privilege.
That's awesome.
So I think, as a field, these technologies, people jumped on them, right? These are amazing. We can do things we never did before. And so as a field, the first thing we all did, sequence lots of stuff and see what was there we never grew in the lab before because it's hard to do, right? And then you kind of hit the brick wall of, okay, so I've got correlation of this with this. Is actually meaningful. And, people would repeat these studies and actually didn't repeat very well. Different bugs were popping up that didn't pop up in your study. Reproducibility is poor as a whole in this sort of study for the field. And so I think the interesting thing for me being in the field for this length of time is that there was this big rush to the sequencing technologies and I did it, perfectly sensible. But now there's a gradual, maybe we need to go back to the lab and do some experiments again and actually work with the bugs and get to mechanisms. And for me, the combination of the two, the high throughput, new modern sequencing methods in combination with actual mechanistic stuff in the lab is what's going to get us to these sort of situations, hopefully, that you're talking about.
And how much further has the sequencing gone in terms of technology? Like, what does it cost now? How fast turnaround?
There was a big dramatic drop off, and it's not been quite as dramatic for, say, 10 years now, but it's still, honestly, if you want to profile someone's microbiota now, it's cheaper just to do the sequencing than do the DNA extraction and the PCR steps involved. So it's like 10 quid to get someone's 16S profile done. It's really cheap. So it costs more to do the extraction and the PCR.
Can we talk about the sort of analytical toolkit that we have? So you're working in the space. Actually, maybe just talk about your research objectives first, then we can talk about the analytical toolkit. I think that's quite cool.
I've got fingers in many pies. I guess ultimately I'm really interested in functions of microbiota that have relevance for health. So we do a lot of work on trying to identify species that are most likely to kill off pathogens in both humans and animals. And so can we harness those to try and reduce carriage of, say, salmonella in chickens? If they've got a strong, robust microbiota that kicks these pathogens out, they don't get sick. And then when we eat them, we don't obviously pick up salmonella from them either. So trying to reduce carriage of pathogens is a major aspect of what we do. And we've found lots of nice bugs, which at least in the lab, will knock out these pathogens very nicely. It's whether they do them in the animal is the next phase.
You will feed them to the chickens?
That would be the aim eventually, yeah.
That's cool.
Yep. I guess we might touch on this later if we're going to talk about probiotics, but
we will.
It's easier said than done, unfortunately, with some of these bugs. So we do that. Very interested in the impact of diet on microbiota. The Rowett Institute, where I'm based in Aberdeen, for years has done lots of really nice work showing how different types of fibre will promote different types of microbes. So I think we have a decent handle now. Let's say, like I say, if you eat starch and you've got Ruminococcus bromei in your gut, that will go up. What we've got less of a handle on is what is the impact of that on the host. So what does that bug do then? Is it a good thing that this bug goes up or is it not? And so for me, I'm really interested in trying to get towards functionality of some of these bugs and working out, which are the better ones and which are not.
Could you please allow me to actually give you and the Rowett a shout out on this podcast? Because well, for a couple of different reasons. One, you've been on a winning streak in terms of awards.
Yeah, no, it's been quite a
which has been cool. Last time I saw you, we may be both getting an award.
Yeah, that's right.
Yeah, so well done to you. Well done to you. Well done to you.
Back to us.
Well done to you. And your group was given an award by the university for your research output. But perhaps, and I'm not trying to sort of discredit or put down the University of Aberdeen at all, you were given one that may, in some people's opinion, be more prestigious, which was the Nature Publishing Group.
Yep.
And your laboratory, or
we only came second, to be fair. I should give it.
Yeah, but this was a global assessment.
Yeah, we did
of
second in the world's not bad. Yeah,
to Jeffrey Gordon and co, which I think is fantastic. So I wanted to just give you kudos for that, but I also wanted to say, sort of on record, because you were one of the first people I spoke to about my interest in the microbiome 7 or 8 years ago, that I do genuinely believe that if it wasn't for your interest and your altruism in trying to help me, likewise with Petra and everyone else who was involved at the time, would never have got going because I didn't know what I was doing. I still maybe don't. No, I'm joking. I know more now. So thank you for that.
No, it was an absolute pleasure. It's been
Thank you
amazing.
for that. It's been awesome, and we've collaborated on various different things for the last 5-7 years.
It's been wonderful to see the company grow. Yeah.
Thank you. So back to the Rowett then and the diet. So I've asked you to describe the sort of analytical toolkit. So how are you using technologies to help answer your questions?
So there, as technology has evolved over the last 20 years, that has also evolved. So initially what we used to do would do sort of marker gene sequencing, it's called, and that's just one gene from the bacteria, which all bacteria have, and by sequencing that gene, you could work out which bugs were present. So that's, like I say, these sort of gold rush days stuff that we did lots of, in disease X, what's in someone's gut versus a healthier control. So that was done for a lot, and it's still done, and it's still a useful thing to do. If you want to know which bug's there, it's a very good way of doing that. As the technology's evolved and it's got cheaper and higher throughput, then what people have evolved to do is just basically brute force, just smash up your sample, sequence as much of the DNA as possible, and then what you end up with is essentially like 1,000 little jigsaw puzzles.
Okay.
These are the genomes of these bugs, which are the tiny little fragments of their genes, which you have to try and put back together. And so imagine 1,000 jigsaw puzzles being dumped in a big pile, and then someone takes the box away, and then you have to try to assemble. That's essentially what has to happen.
Okay.
And so there's been huge revolutions in the computer algorithms to try and do this sort of mathematical problem solving to put these together. And we're at a point now, which I think is super cool, is that you can basically construct a genome from a bug by piecing together all these little bits of the puzzle that you've never grown in the lab before. And essentially you have the complete biology of the organism there in its genome. Wow. So in theory, you should know what that bug can do. In practice, bugs don't always do what their genes say they might. Just because they've got the gene doesn't mean they're actively doing anything with it. But in theory, that allows you access to any bug, as long as you can sequence it in enough depth that, like I say, we maybe haven't grown in the lab before yet, know nothing about. So
just on that
super
point,
exciting.
That is super exciting. But just on that point, why is it important to have grown something in a lab before? And how much of the microbiome within the human database has not yet been cultured?
So good questions also. I mean, I do a lot of culturing work, so I'm going to argue very strongly for culturing, but I can back it up with reasons. So if you read the literature, one of the things you'll read quite often is that 99% of bugs can't be grown, and so we need to do sequencing. That might be true in things like soil, when bugs double every 3 months. You're never going to grow that in a lab. In the gut, we're quite fortunate. The bugs have to grow fast, or you're going to poop them out the other end. And we kind of know what they're eating, right? They're eating what we're eating. Right? So, So actually, a relatively high proportion of the bugs, maybe 60, 70% of the bugs in the gut have actually been grown, we think. Maybe more, depending on some of these genomes turn out to be real and not artifactual, but I won't go into that. So we actually have a pretty high proportion of the bugs from the human gut already in culture. And the reason why that's good, there are multiple reasons. One is actually, if you have it in the lab, you can do experiments with it. A DNA sequence you can't really do much with other than try and work out what it does. If you have it in the lab, you can test it. So say, this bug has the genes for starch utilization. You can grow that in the lab and test it. And sometimes what the genes say it might do doesn't always match what it does. So actually verifying things is good. You can use those bugs to make much better reference databases. It's a bit technical, but essentially the better the reference databases, the better the sequencing-based projects are. You can map them back so you can work out what the bugs are. Having a name for a bug can actually be super useful. A lot of people don't like names, but if I tell you you've got C. difficile in your gut, I think it's useful to have a name for that bug rather than sequence number XY53.
I totally agree with that.
So having a bug in culture means you can characterize it properly, give it a name.
It also allows you to bestow an honor upon people who do good work, right?
That's true, yeah, you can. You can give a few people at Rowett who've got bugs named after them now. Not me, sadly, but So
what do you have to do?
Just hang around and be good. Honestly, it's probably one of the highest honours you can get as a microbiologist.
For sure.
That someone's valued your work enough that they'll name the bug after you. And I say at the Rowett, we're really fortunate. We've got Petra, Sylvia, and Ari who've all had bugs named after them.
Is that right?
Which is really amazing, actually. Yeah. It's a testament to the great work
That's
they've done.
class.
Way before I started, they were doing fantastic work. So
that's class.
No, they're brilliant. And the final thing, the good thing about having bugs in culture is, if you have a bug in culture, you can develop it as a therapy. Right. So, if the bug turns out to be a beneficial type probiotic type bug, you can't give someone the DNA and hope that it's going to do anything. You need the bug. So for all those reasons, and that's why I talked to you before about the shift in the field from lots of sequencing and now a sort of gradual realization that maybe we do need to go back to the lab a little bit for those reasons. There's lots of value still in the old school microbiology.
How is, to use a buzzword, AI
Yeah.
coming into this field?
Yeah. So, it's a really good question. And it's not my core area, so I'm kind of speculating a little, but it's fine, I'll speculate. So, given the complexity of the microbiota, right, it's too much for the human brain to try and disentangle. So I do think there is potential there if you've got a computational brain doing the work for you. The only note of caution I'll add is that, of course, modeling and this sort of stuff is only as good as the data you feed into it.
Right.
Garbage in, garbage out.
Right.
So again, if we're going to talk about myths later, if you type into ChatGPT who invented the word microbiome, it will tell you it was Joshua Lederberg, and that's not true, right? But because ChatGPT reads this in all the literature, it parrots back what it's been fed in. So AI can take you so far, but fundamentally you need that high quality underlying data. And as we maybe talk about, it's not always the case with quite a lot of microbiome.
Have you tried to debate with ChatGPT on something?
I have, yeah.
So have I. I was really impressed. We had a long debate around full spectrum versus defined consortia.
Yeah.
I was like, keep going. It's really good.
Yeah, no, honestly, I think
really good.
Yeah, no, it's it can really hit the spot sometimes. And again, I think it's down to the quality of the data that it's given.
That makes sense.
Sometimes it's off. For a laugh, I typed in, is Jim Goodwin a good manager? And it said yes. That's wrong. Right. So that's an esoteric joke for Aberdeen fans there. Anyway. It's like I say, with these tools, they're only ever going to be as good as what they've been fed on, and that will obviously get better as knowledge improves and it adds to it. So I do think it's a really exciting area, and I do think it's got huge potential particularly for really complex problems like the microbiome, where it's just so difficult for a human brain to disentangle.
Now, you've spoken about marker gene surveys, so 16S sequencing, and that brings me to something I wanted to discuss with you, which was at-home microbiome testing.
Yes.
So it's kind of fascinating, right? So you're someone who's been in this field for 20 years. You're part of a research group that's been given the highest accolades. And you said earlier on that you can't tell what a healthy microbiome is by looking at it, right?
Yeah.
So there's lots of companies offering this. I'm not going to name names.
That's good.
But we both know people who are involved in this.
Yeah.
How much value do you think these at-home tests have?
So I think the answer I usually give, because I get asked this quite a bit, is that, sometimes it's just nice to know. So I've been involved in studies. I got my microbiome profile. It's quite interesting to see what's in there, right? I've got these bugs. I've got these bugs. I think, as I alluded to before, the problem is we actually don't know what the vast majority of these bugs actually do. And a lot of it's probably context dependent anyway. A bug that might help you protect you against pathogens might also be pumping out a chemical that might increase your risk of bowel cancer 30 years down the line, right? So is that a good or a bad bug, right? And so I think the problem is that even something like E. coli that people have studied for like decades and decades, we still don't know what half the genes do. These bugs, which, really are massively under-researched, and even now with the big activity in the field, we really don't know what the vast majority of these bugs will do. And that's what really fascinates me as a scientist. That's the stuff I'm interested in. So when you get a profile from these companies, I guess, if I had low diversity, lots of proteobacteria, I'd be like, that's not great.
What would you do?
Yeah, but I'd probably start eating more fibre, but I guess we come on to that.
Yes.
But I mean, does that tell me fundamentally that I am definitely screwed with something? It doesn't, right?
No.
And I think that is the issue. I think a lot of people who do these tests, they're kind of worried and they've got concerns and they do it and they hope that the answer will be in their microbiome. I know the answer might be in the microbiome, but unfortunately where we are right now, it's very difficult to take a profile and go, right, you've definitely got this problem X, Y, and Z. I mean, even somebody, if you've got C. difficile in your gut, 3%, 5% carry that without any harm done until they're old and they take antibiotics. So
babies have loads, right?
Exactly, yeah.
Babies
exactly.
have tons of C. diff.
Exactly, yeah, and they're fine.
Yeah.
So again, just because something is on a profile doesn't necessarily mean that you've got something bad. So that's where we're at with the microbiome field. I think the answer to a lot of questions you might ask is that we're kind of in a vacuum situation now where people know the microbiome is important. It is, we've discussed this.
Yes.
But we haven't quite worked out yet how to harness it. And so that vacuum is kind of filled with sort of traditional probiotics, these sort of services where they're interesting, but actually the scientific proof at this point, possibly not there in a lot of cases.
On your statement around most people believe the microbiome is important, I would concur. It's changed a lot in the last 7 years.
Yeah, people definitely are more aware of it, yeah.
Still people, in my line of work, I'm involved in trying to convince people that this is important and it's worthwhile investing in, either as a partnership or through an equity investment or whatever.
Yeah.
I do still come across people who say, it's not important, highly skeptical.
So, I mean, I'd like to think the field has moved away from that point where people are skeptical about its importance. I think you can have skepticism for individual conditions, right? So, like, you know, my one is obesity, right? I don't think the microbiota is a major player in obesity, right? If you want to lose weight, you know, the traditional ways of doing that are going to work way better than taking a probiotic, right? And I'm happy to discuss that.
Yeah, because this is a debate I've had with three people.
You know, I have skepticism for individual conditions, right?
Yes.
The people who write off microbiome for everything are being contrarianism for the sake of it. There's lots of promise in the microbiome. Whether we're there yet in most conditions, I would argue probably not, but the promise absolutely remains.
Can we talk about obesity?
Go on then.
So I had a debate with Prof. Karen Scott on this, and I also had Richard Hansen and Kostas from Glasgow and Dundee. Well, Hansen's now at Dundee.
Yes.
Kostas is in Glasgow.
All great people. Yeah.
All great people.
Yeah.
And I'm just really here to be devil's advocate. You know, I believe fundamentally that the best way to you know improve your body composition is a regular exercise regime, looking at your macronutrient intake, changing your calorie intake if it's too high.
Yep.
I don't believe a calorie is a calorie.
Okay.
I mean, it is insofar as it is a unit of measurement.
Sure. Yeah.
However, if you're giving 100 calories in a bar full of fiber versus 100 calories in dextrose tablets, your utilization of those calories is going to be very different in the body.
You will utilize them differently.
And by the way, fiber is huge.
Yeah.
Fiber is fundamental, which is why you can eat loads of fruit and not see dramatic shifts in your blood glucose, and fructose is okay in fruit because of the fiber.
I'm going to get most people.
Most people.
There's a brilliant study that came out, I think it's 2019, Zevia et al. It's in Cell, where they talked about personalized nutrition, and they did the glucose
Israeli
monitors.
group?
Yeah, they did the glucose monitors on the arm. If you can get one of them on, you should talk to them. They're interesting. And it's a really interesting
can you
study.
link me?
So,
do you know them?
So yeah, I can.
Yeah.
But anyway, so
Cool.
But yeah, no, it's super interesting. So they took a bunch of volunteers and they put them with these continuous glucose monitors, so they could track blood glucose all the time. And they fed them set diets, and so then they monitored how their glucose spiked. And so for most people, you give them like a biscuit, a cookie, you get this spike in blood glucose, as you'd expect, right? You give them a banana, pretty flat, as you were just saying, fruit, fine. For a small number of people, the opposite pattern happens. So you give them a biscuit, a cookie, no blood glucose spike, give them a banana, boom. Right, so at a population level, the advice, eat bananas, not cookies, is excellent advice, right? But at the level of that individual, that's terrible advice. You're basically getting them to eat something that's continually spiking their blood glucose levels. So, and I think, you know...
Is that microbiome at play?
So they argued a little bit. I argue possibly, I don't get that, to be honest, because most of the sugar is absorbed in the small intestine before it even gets to your colonic microbiota. So what's in your stool will not at all reflect what is utilizing sugar further up in your gut. So I have some dubious thoughts about that. But anyway, but you're right about calories not always a calorie, but I do think how the body responds is probably a bit more individualistic than has been considered in the field before, and it is an area that people are going into.
And the microbiome could be a determinant.
So the microbiome and obesity, I mean, if I'm going to touch on that, I'm not saying microbiome has no effect on weight, right? It's clear if you have a microbiome that's better able to extract energy from your diet, you will get more. And the reason that happens is it breaks down the fiber into short chain fatty acids. You absorb them, you get energy from those acids. Now, some people's microbiota will be better than others at breaking down and releasing sugars. So acids, sorry. So there's definitely going to be some amount of variation in the amount of calories you extract from fiber, for example.
Yep.
But you're talking tens of calories, not hundreds of calories, right? And so over the course of your life, people say, yeah, but that tens of calories every day adds up and adds up and adds up and adds up. Your body will equilibrate, right? A larger body needs more calories to maintain it. So I can get how your microbiota might move you to a little bit and then you equilibrate. It's not going to continue and make you more weight, more weight, more weight, more weight. So no, I don't believe that the microbiota is leading to an obesity epidemic. It hasn't changed that much in 30, 40 years, whereas the levels of obesity has really rocketed.
Is that true though? Because the food industry, in some respects, has moved to a much more calorie-dense, micronutrient-light, fiber-light composition.
Definitely
And I'm not... Where I sit on this actually is more our diet's changed, so our microbiota's changed.
Yeah.
And the microbiota, through mechanisms, and I'm getting a bit into woo-woo land here, but just let me
feel free,
indulge. I think there's so much you don't understand about the communication between the microbiome and the wider body, including the brain.
Yeah.
And my view is that, so it's a self-perpetuating cycle of, I'll call it badness. It's not a scientific term, but it's badness.
Yeah.
So the cycle is, in someone that's has a predisposition through genetics. And I believe that obesity is largely genetic. It's human.
Man, I agree with that. I got my DNA sequence, just to butt in briefly, I got my DNA sequenced when I was at the Sanger Institute as a sort of pre-test, and I got it back, and it was like, you're bald, you're very pale, and you're probably overweight.
Is that what it said?
Yeah, I didn't tell it that, right? So that
is that what it said?
That's what it said, yeah. So, there is something in that. People will eat more depending on their genetics.
That's fascinating. Yeah. So when we were asked about what does Alan look like, I would have said, just look at his genome sequence. And that will tell you exactly what you need to know.
Nailed me right down to it, like red hair.
Amazing.
Yeah. And like, no, I told them nothing about me, obviously.
That's fascinating.
So part of it is in your genes.
Definitely. But can I just go back to the badness?
Please, finish your point. Sorry.
The badness and the self-perpetuating cycle. So someone has a predisposition. Now, if that person was in a jungle and all they had to eat was fibre and vegetables and lean proteins, I'm not sure that they would be as obese as they were.
You're 100% right. Environment plus genetics interacts.
And the last element is, so yes, it's the calorie-dense foods, but this self-perpetuating cycle, just to wrap this up, is the microbiota changes in response to the input, which is these sort of sugary, maximally processed fibre-like foods.
Yeah.
Therefore, you may get these blooms in, for example, the proteobacteria, Enterobacteriaceae, that kind of thing.
Yeah.
And through mechanisms that I hope you or someone in the realm of research determines, they communicate with the brain and they say, we need some more of that.
Okay.
We need some more of that sugary stuff.
I think the brain already asks enough for that sugary stuff. I think they need your microbes on top.
But is it
tastes good?
It tastes good, but maybe there's something more there. Could there be something more there? So the small intestine goes, we just love this sugar bath. We need more. So we're going to produce these molecules.
So I will say I'm not a scientist that works on the biology of obesity, but I work in an institute that is, obviously. So you pick up some knowledge by osmosis. The body is a brilliant, fantastic feedback loop system. It detects when it's eating food. It releases all sorts of hormones that determine whether you want to eat again or not, right? There's certainly some evidence that some of the things the microbiota can make might cross-react with hormones, for example, like GABA, serotonin. So I get all these things, right? But again, whenever you try and do things like fecal transplants in obese people, they do not lose weight. You do things like insulin, things maybe happen. They know this has been done multiple times. No one's losing weight on a fecal transplant.
I have also looked at this evidence extensively, and my issue with it, well, I have a few issues with it. One issue... I have many, many issues with fecal transplant studies in terms of, is what you're giving to each of the patients the same? Is the study properly designed, so on and so on. It's very hard to make the case for FMT in this particular indication that FMT alone, without controlling all the other factors like diet, for example, is going to have an impact. But
it's minuscule,
Yeah, so, but you're right. So this whole thing started with the case report, I think it was 7 years ago now.
Yep.
You know the story?
Yeah, I know the story, but please, probably your listeners don't. So,
Well, I'm sure we've talked about it before, but there was a case report, high impact journal case report. Essentially, a woman received a microbiome transplant, we call it a fecal microbiota transplantation or an intestinal microbiota transfer, to use the updated nomenclature, from a relative who was overweight. And this case report described rapid onset weight gain.
Yeah, sure.
So everyone then said, okay, microbiomes caused this, but then there's been randomized control trials since, there's been retrospective analysis of stool bank studies, and there hasn't been any impact.
So again, I cannot get across to people who may not be scientists, general public, that case reports are the lowest form of evidence, right? It goes back to, I can take a crystal from a crystal shop, rub it on my rash and my skin, my rash went away, the crystal did it, right? Right, there's no evidence for that.
It might have done
it. No, it might have done it, but then but it didn't, right? So that person may have gained weight anyway. There's no proof that came directly from the transplant that they got. So case reports are interesting, and they might send you down a path of inquiry. But on their own, they are worthless almost. Again, I think we talk about this, maybe just briefly detour into the scientific method. The scientific method is beautiful, right? It's science gets a lot of bad press, and scientists are terrible people, blah, blah, blah. Scientists are human beings, they make mistakes, right?
Yeah. But that's
a very small proportion are fraudulent, right? But the scientific method is brilliant because mistakes get corrected, right? And so a case report is a case report. Does it repeat? Do other people independently find the same thing? If the answer is no, then that's a weird oddity that who knows what happened, right? It's the crystal on the rash, right? It's nothing. And so the scientific process is a beautiful thing. The problem is it's really slow, right? It takes years and years, decades and
Can I
decades.
just ask you a question? Are you into crystals?
No.
It was just a random thing.
I see crystal shops in my local high street and they really annoy me. But anyway, that's
I didn't have you down as a crystal guy, but I just
quite the opposite. Yeah. I just don't like. I don't like woo.
Yeah.
I'm a scientist by nature. Anything you claim should be evidence based, and
Got it.
And so if that case report lacks the level of evidence required to really claim that, that's what caused it.
So I agree, and I'm just stirring the pot a little bit. To see
Stir away.
Now we've talked about diet and the microbiome.
Mm-hmm.
So one of the questions we get quite a lot from the listeners is, what can I do to improve my microbiome?
Yeah, I get that a lot too. Yep. So I mean, like I said, I'm a microbiologist for 20 odd years now. The only thing I've ever done to improve my microbiome is eat more fibre. I don't take probiotics. I don't do anything else because essentially I don't need to. Apart from being a bit chubby, nothing wrong with me, right? It's not going to make my hair grow back. But I do eat more fibre. Because there's lots of evidence now that, for most people, and I really, it's a nuance again in microbiome, definitive statements are never true. For most people, more fibre is the best advice you give them at this point in time. And that has the nice benefit that it basically agrees with nutritional advice going back decades, right? That fibre is generally a good thing for all sorts of other reasons, right? Cholesterol lowering, improves transit rate, whatever. There's a million reasons why fibre is good. And so what the microbiome work does is just add another layer to that recommendation. Yeah. It doesn't tell you anything different. Now there are some people, some of the mechanism. It's not going to be all the mechanism, but it'll be some of it. Now, for some people, unfortunately, they just cannot tolerate fibre, and so for those people, it's actually bad advice. You can give them IBS. And so again, it has to be nuanced. For me, I don't get IBS. I'm quite happy eating fibre, and so the one thing I've done since becoming a microbiologist is I do eat more fibre regularly. I was a very Scottish male, lived off.
What does that mean? Bacon rolls and chips,
butteries, rowies if you're from Aberdeen. No one else knows what that is. A lot of just simple dishes, no pasta, no vegetables, no fruit. Just beer. Scottish man. Since working on microbiota, I do eat a lot more fibre than I used to. Yeah.
So, can we unpack that a little bit? So when we talk about fibre, can we break it down? Soluble, insoluble.
Yeah. So you're right. There's lots of different types of fibres. Different plants will have different types in them. Some fibres will feed particular bugs, other fibres will feed other bugs. And so the general advice usually is a nice mix of fibres to try and give everyone a bit of something that they like, in their gut. The interesting thing for me from fibre is, you can take an identical fibre, you and I can eat exactly the same fibre, but have a very different gut response. And the reason for that is that you have a different starting microbiota than I do. And so say again, we go back to that example of starch. If you've got Ruminococcus bromei in your gut, it will go up. We've shown that repeatedly now. If I don't have Ruminococcus bromei, and not everyone does, something else will go up instead. And so say I've got Eubacterium rectali, that might go up. Now Erectalia makes butyrate, Ruminococcus bromei makes ethanol. So, is it a good thing for you to take resistant starch if it makes an ethanol producer go through the roof? Maybe, maybe not. We don't know what Ruminococcus bromei does for health. For me, in theory, a butyrate producer, that should be good to take resistant starch. But maybe Erectalia makes other things that are nasty and increases your risk of bowel cancer 30 years down the line. And that's what I'm getting at when I say we really lack, at this point in time, the ability to say with confidence that if you do intervention X, Y, Z, this is what's going to happen to you. Because a lot of it is quite individual.
What I found really interesting observing science and observing people who are interested in gut health. So it used to be this sort of naturopath, functional medicine doctors. If you go back in time, they would say things like minimally processed foods, eat lots of fibre, don't consume lots of sugar because it damages the gut barrier integrity. And that was woo for a while, but now it's not woo and it's in vogue. And it aligns to what you're saying though.
So again, I think some of the reasons behind it are woo. So I think the general advice is fine, but you get for example, you can Google up things online, eat cucumbers, it scrapes the bad bacteria. Bacteria is put off.
Does it do that?
No, it doesn't, right? But that's woo. But eating cucumbers, fibre, is probably not a bad thing, right? So I think I would warn people that a lot of these general good pieces of advice, although they are good pieces of advice, the evidence they use to back that up is guff. It's nonsense. Yeah.
How do people know if it's guff or not?
So this, for me, is the biggest issue in the microbiome field, right? You can Google up microbiome right now and you will be inundated with a million different things, telling you a million different things that they'll tell you have brilliant evidence for. And I guarantee you, almost without fail, most of them will have no quality evidence for the claims they're making. And so as a consumer, what do you do? It's...
What do you do?
Well, I don't have any of them. That's what I'm saying. As a consumer, I eat more fibre. I don't take anything else, right? But again, I'm ostensibly healthy. I don't need to, in my mind, take anything else. People get desperate. They've got conditions. Their normal doctor is not able to help them, so they go to the internet. I absolutely empathise, right? You'll try anything. People take advantage of that, There are people making a lot of good money off the microbiome without much evidence to support it, unfortunately.
How do people know what kind of fibre they should be eating, or how do you know if, an apple contains pectin?
Yeah.
Right? Chicory contains inulin.
Yeah. Yeah.
But what does that mean?
So again, like I said, it depends on me or you who's eating it, right? If you've got a decent pectin degrader like Faecalibacterium prausnitzii makes lots of nice butyrate, then for you eating apples might be great. If I've got a Bacteroides uniformis that eats lots of pectin and makes a nasty sort of compound, maybe it's not great, right? So again, the problem is I cannot reproducibly tell you have to have fibre X to be healthy. I don't think I'm there yet. Now, some people are trying to do that, and I think this is an area that is actually coming rapidly.
It's coming.
But right now, it's very difficult. So again, if you eat an apple and you don't get sick, you don't get bloating, you feel fine, keep eating apples, That is my general advice. If you're happy with it, you feel good with it, fine.
So people should try lots of different things, figure out what works for them.
For me, yeah. Yeah. I mean, the nice thing about fibre is, unless you're one of these people that can't tolerate it, it's unlikely to do any harm, right? So, yeah. So even if you're not getting any benefit from it, it's not going to do you much harm.
Tell me about probiotics.
Okay, yeah. So, I mean, I'm gonna rant, sorry. Yeah. So I think... probiotics as a concept is a great concept, right? Taking a beneficial bug, improving health. There's lots of nice promise in that. The word probiotic itself, I hate the word probiotic, right? And I'll tell you why I hate the word probiotic. It's a meaningless word, right? Because what is a probiotic? If you think of probiotic, what is a probiotic? You tell me, name me a probiotic.
I would refer to the WHO definition.
No, I'm talking about a bug. Name me a bug that's a probiotic.
Faecalibacterium prausnitzii.
Okay, you can't actually buy that as a probiotic, but anyway, name me another bug.
Was that a probiotic bug?
No, you can't buy them in the shelf as yet, as far as I'm aware of. Name me another probiotic.
That'll
bug.
be my next venture.
Yeah.
Coming soon.
Well, we can talk about that in a minute.
Coming soon.
Well, you go to the supermarket, what do you buy in the shelf?
Okay, so supermarket ones, we would have lactobacillus, we would have bifidobacterium.
Okay, so I'll stop you there, right? So you've named me two bugs from two completely different phyla of bacteria. They have not been related for billions of years, and yet you've called them the same thing, a probiotic, right? They are totally different things, right? So to me, as you've got a clinical background, right? It's a bit like saying, you've got a headache, mild headache, I'll give you heroin. It's a painkiller just like aspirin, right? They're both painkillers. That would
probably do the job.
It would do the job, right? But you might have some downstream problems, yeah? So heroin and aspirin are both painkillers, but they have very different like mechanisms of actions and problems, etc. So you would never do that with any other type of intervention. It's like, oh, you're ill, I'll just give you a drug. Which one? Right? So probiotic is a meaningless word because there are so many different bugs with so many different activities. What do you mean by probiotic, right? And so the word is highly unhelpful. And again, even if you get down to the species level, you've got a lactobacillus, whatever, in this pot, and you've got the same lactobacillus in this pot, but it's a different strain, right? We had a PhD student called Matthew Dalby used to use this analogy, so I'm going to steal it from him. Rottweilers and Chihuahuas are both dogs, same species, but you're only using one as a guard dog, right? Say Chihuahua, they're vicious, right? But they're both dogs, right? Same species. These two lactobacillus in the pot are both the same species, but they're different strains. So, different strains can have very different activities. So for me...
Can we exemplify that with E. coli?
Yeah, well, E. coli 0157 will rot your kidneys and potentially kill you. E. coli nisli is a probiotic, right? It's the same species, right? So for me, it's just a not helpful term. It's if you need the precision of something to work, you need the precision of the product. And the word probiotic. So, you do meta-analyses. So we looked at the effect of probiotics on mental health, whatever. And you look at it, there's like 10, 20, 30 different products, different doses, different lengths of time, different species. Oh, we didn't find a consistent picture. Well, of course you didn't. You're looking at completely different products. And so the word probiotic is just useless. We need the precision of, if I'm ill with inflammatory bowel disease, I want to take F. prausnitzii, not a probiotic. You want to take the bug, So, and that for me really frustrates me. You can see I'm getting quite animated about it because it really doesn't help the field progress either. Because people as consumers take a probiotic, it doesn't work for them. No, it's all rubbish, right?
Right.
And it isn't all rubbish, right? Some of it's rubbish. We've talked about this, but not all of it is. There's some good stuff in there as well. So
Where do we go next?
Honestly, this is the toughest question you can ask me the whole day. It's really tough. I mean, the bar has been set quite high by the European regulatory authorities. You have to have a really proper health claim. And actually, I'm okay with that because I am a consumer fundamentally. I want proof that my product will work. But that does stifle innovation, because if you've got a product that billions of people buy already, then why would you do the test? I might tell you it doesn't work. Right? So you've undermined your own product. People are buying it anyway. So it's really tough, So where I stand on probiotics is they have huge potential. The word itself doesn't help. I think we need much more precision, one type of bug for one type of condition,
right,
and really get the evidence that that particular bug will do something.
So that sounds a bit like a drug.
Yeah, no, it does. Yeah, for me, people talk about it being a food. I think they sit in that middle ground, right? And it depends what you want to use it for, right? If you want to take one and feel good about yourself, then fine. You don't need the level of, let the placebo effect kick in, be happy, right? But if you want a particular bug for a particular condition, then that's a drug, right? You're trying to cure something.
So can you, with all the experience that you have, talk about how, I'll use I know we don't like the word, but I'll use the word probiotics.
Fine, no, it's fine. Everyone does, fine.
So the currently available probiotics that are off the shelf, how they were designed and developed.
Yeah.
Versus where we need to go.
Sure. Okay, so I will say there is decent evidence for a few of these products for specific conditions that actually they have decent efficacy. So, premature babies are a prime example. Babies are born very premature. They're very susceptible to an inflammatory condition called necrotizing enterocolitis. There's now pretty good evidence that any of these lactobacilli bifidobacteria, you can really drop the rate of that really terrible disease by about half, right? So some conditions, these existing probiotics, actually the evidence is good. For most things that are claimed for them, the evidence isn't there yet, I would say. It doesn't mean they don't do it, but at this point in time, the evidence is usually not there for most other things. Now, I would argue that these are the wrong type of bugs. Lactobacilli are relatively dominant in the small intestine. So if you've got a small intestinal problem, maybe they're the right bug. In the large intestine, they're 0.01% maybe on average. They're a minority component, right? So if you want to solve something in your colon, it's the wrong bug, right? And so we would argue instead that some of the other bugs, like your Faecalibacteriums you talked about, your Rosburias, your Ewicterias, all these bugs, which most people have never heard of, they're the ones that are 5, 10, 15, 20% of your microbiota. So if they're healthy, I would argue they're likely to have more of an impact than these invading type lactobacilli that don't live there anyway. So that's the principle. These are where we might want to go in the future, and lots of people are looking. Again, there's also a caveat with microbiome. One is we still don't really know yet what effects a lot of these bugs will have. Some of them might have off-target effects, might be very individual specific. And also, they're really hard to work with. So they're obligate anaerobes, so they die when you expose them to air. So some of the bugs that I've just talked about, Faecalibacterium, a few seconds exposure to air, it's dead.
Wow.
So you're not going to put that in a yogurt pot and put it on a supermarket shelf. It's just not going to survive. So there are challenges to getting some of these bugs into people. Now, luckily, there's lots of people in various parts of the world, I mean, not lots, but a few people trying to get through that technical challenge. And I don't think it's insurmountable. I think it's definitely plausible that you get some sort of encapsulation that keeps the bugs safe enough that you can swallow them and then get them into your body. So I think that's a nice thing about being such a dynamic sort of active field, is there's lots of progress being made all the time. And so I do hold out some hope that these bugs will eventually be useful as sort of next generation probiotics, if that's the word you want to use. But I do think as a field, a specific bug for a specific treatment is probably the way to go.
So there's microbes everywhere.
Mm-hmm.
Why do we not see microbes that you see in a dog in a human?
Yeah, it's a good question. So partly it's diet. So a dog obviously is not eating much fibre. So, the fibre degraders aren't going to set up home in there. Partly it's host selection. The anatomy of a dog intestine is different from our intestines. So the bugs that like our intestines don't necessarily like what's in a dog's gut. Some of it's host genetics. They will select different bugs. So again, what they're exposed to, dogs eat poo. Most people don't eat poo, right? So you're not going to be inoculating yourself in that same way.
Right.
So, all of these reasons combine to make us sort of more unique as a species with what we've got in our gut.
How many different types of species are there in the human gut?
Yeah, that's quite an open question. So it depends on the method used, So if you culture the bugs, you might get a few hundred. These metagenome assembled genomes I talked about, they're getting into tens of thousands now. So, whether they're true or artifactual, is, these jigsaw puzzles might just be a couple of jigsaws stuck together from different bugs, and it looks like a new one, but actually you've created this weird mosaic. So some of them may not end up being real. But yeah, so I guess the open question is probably at least a few thousand, I guess, based on the evidence. And in each person, there'll be a sub-selection of those. And, there's ones that pass through. There's microbes in your food, you detect them in stool, but they're not living there. They're just passing through, things that you swallow in your mouth, all these things. So, what is a true gut colonizer is not always what's in your stool sample. Some things are just passing through.
And what about genetics and genomic diversity? So within the human genome, it's quite constant, isn't it?
We're all
20K.
Yeah, we're all very, very similar to each other. Yep. Cosmetic differences only. Yep. And the microbiome came up to 50% different. So it's a much more diverse I guess, phenotype that you get as a human being in your gut microbiome compared to your genome. So it's one of the reasons why the microbiome is considered quite a druggable target. There's not much you can do. I mean, things are changing with genomes, with CRISPR-Cas type technologies and things, but in theory, it's easier to tweak a microbiome than it is to tweak a genome. So and one of the reasons why there's such an interest in using it therapeutically.
We've talked so much about bacteria today, but it's not only bacteria, is it?
Yep, no, it's true. Yep, you have a small number of fungi in there relative to the microbes, the bacteria. Sorry, and they have some impact on the host via their immune system and things as well. You have a load of viruses in there, an absolute pile of them. I will say that most of them are not the kind that we have to worry about. They're the kind that the bacteria have to worry about. So most of them are in there infecting bacteria. So. It's one of the amazing things of microbiology is you get these tiny little things and you've got even smaller things that prey on them. So most of the microbes in the gut are basically, the viruses, are attacking the bacteria, not us. Now that might have implications for our health because if it's a beneficial bacterium and it's been wiped out by a virus, then that potentially has impacts for us as hosts as well. But I have to say that's much, much, much less studied than the bacterial component. And viruses are tiny, much more difficult to work with. So yeah.
And the listeners should look up these phage bacteria because I just think it's so cool. It's like little
the biology of that is mind blowing. Yeah, they dock. Yeah,
they dock and then they
steer. Yeah,
it's so cool.
They are they're like something out of a horror movie. Yeah, they just come in with their little tentacles, grab onto the poor little bacterium, and then inject their DNA through it, and then basically just make that bacterium make lots and lots of copies of the virus until it gets full, and then it explodes and the virus comes out. It's like it's something from Alien, right?
Yeah, it's incredible.
Truly, nature is both horribly cruel and also horribly amazing. Yeah,
it's happening inside of us right now.
You've got basically inside your gut right now, your microbes are breaking down what you had for your breakfast. They're all fighting with each other for it. Some of them are getting on together as friends. It is like a city, the same as humans interacting right now.
It's amazing.
It's amazing, yeah.
So where do you see the field going then?
Yes, I mean, where the field is going, I think it's already on this path. It's moving away from the sort of correlative stuff towards more mechanistic stuff. And I do see it coming towards products that actually do bring benefit to health. So, I think we talk about C. difficile a little bit earlier in the day. I guess I'm 44 now. So, by the age I'd have to worry about C. difficile usually is a little bit older in life. When you're a bit more elderly, you're in a hospital bed on antibiotics. I'm pretty sure I can rule out C. diff as something that's going to kill me, right? Because I think by the time I'm old enough to have to worry about it, we will have proper treatments that will do the job. So I think that's one example where, I absolutely believe we will have microbiome therapeutics that work reproducibly and are used in clinical practice. I think the promise is too high for that to be ignored. There's too much invested in it now. It's coming.
Yep.
How far that goes, I guess C. diff is a low-hanging fruit. You've got one pathogen there, you kick it out, the problem's solved. Some of the other conditions where microbiomes be linked to are much, much more complicated. Yep. There's underlying host biology in there to be dealt with too. There's damage to the body already. And so how far you can take that microbiome, who knows? But it's super exciting to be in the field trying, right? So, no, I think honestly, next couple of decades, I don't say next 5 years, I'm talking decades. I know some things go fast, but usually scientists, you are more optimistic than me. I just published a paper from 13 years ago. Right?
What?
Yes. So I did the work 13 years ago. So that went past like that. So when people say, where do you see the world in a decade? If you asked me 13 years ago, I wouldn't have expected I'd still be writing that paper. Yeah. So, the world goes faster than you'd like, unfortunately.
I describe it as pathologically optimistic about everything.
You certainly are, James, but it's not a bad thing. Yeah,
about everything.
Yes.
Timelines, technical feasibility, what a small group of team members can do within a time frame, but that's
given the
quintessential-
motivation, the funding, the impetus, people can do amazing things. The issue with the microbiome is people are having to do amazing things against a very complicated backdrop that we've talked about, so it's like scaling Everest and then scaling another 100 afterwards. It's a tough challenge.
I'm with you.
Yeah. So
can we talk briefly, about FMT? I know we said we're going to move on, but you're here now, and
why not? Yeah.
So, open question then. What are your thoughts on FMT?
Yeah, so again, it's nuance always. Every time I answer a question, you never get a straight one from me. For me with FMT, it's very much a risk-reward intervention. So if you consider C. difficile, where you've got a very elderly patient usually, who is probably going to die without the FMT, then the risk is very low, right? That poor person was going to be very ill anyway. They're very old. There's very little chance for anything to happen bad after that, right? The reward is you cure the disease and the person gets better. They leave hospital the next day, right? So the risk reward is very much in favor of fecal microbiota transplants in that specific case.
Absolutely.
If you take something else, again, I'm not going to give examples here because I don't want to encourage these sort of things, but there are people giving fecal transplants to children, for example, for conditions where the reward is not clear because it's not actually been proven that microbiota transplant will do anything beneficial for that kid. And the risk is that, 30 years' time, you give them bowel cancer.
Sure.
So for me, the risk reward doesn't stack up there. So the nuanced answer to me is, what does the evidence tell you, right? If the evidence supports its use and it's relatively safe and the risk-reward balance is right, then why would you not do it? If the evidence isn't there, I would say not yet. No, you need more proof.
I'll give you a thought experiment. If I could produce a capsule for you that you had an extremely high degree of confidence that there were no pathogens in there,
Yep.
and it had high diversity,
Yep.
and it had 20 different or 50 different functional groups
Yep.
associated or genuses that you would commonly associate with healthy people.
Yep.
Is there ever a scenario where we would give that to people just routinely to sort of
It's a really good thought experiment.
because and I'll bias you a little bit perhaps. Or I can't bias you, but I'll tell you my thoughts. Just
Go for it, yeah.
I do believe that will be a thing. And I want to try and make that happen.
I don't think it's beyond the realms of possibility. I think, as we've discussed, you can take that pill and you might have a diet that supports them and they'll stay in your gut.
Right.
I might take that pill and I go back to eating chips and they all die off anyway, right? So it was pointless for me. So again, given that same pill isn't going to have the same response for everyone.
With you.
What the person does is going to have a major impact on how that pill does.
Yeah.
The next thing I say, it's a bit like, this is probably going to be completely obscure for anyone who's not listening in the UK. It's a bit deal or no deal. You've got your box, you choose to open it or not, right? I've got my box of bugs. Do I stick with what I've got or do I take your one? Because I don't know if what you give me in 30 years' time gives me bowel cancer, right?
That's interesting.
Or if my one gives me bowel cancer in 30 years' time. So it's a deal or no deal. You're taking a leap of faith.
Right.
We don't have proof yet of the long-term impacts of these bugs. So some people, my assumption is people who are not quite happy with their gut and have issues, they would probably take it.
Yeah.
People are happy, probably I'll stick. Yeah.
Would you take it?
I'd stick, I'm all right. Yeah.
Are you sure?
Yeah.
Even if it contained all the good ones?
Yeah, I told you what's good, what's bad, context dependent, right?
Now, you mentioned bowel cancer a lot.
Yeah.
Why is that?
It's one of these examples where there's microbiome plausible links, but it's one of these longer term things. It usually hits people older in life, right? Okay. And certainly the way the microbes function in the gut, break down products of proteins, for example, they're known carcinogens. Some of them make butyrate, which is an anti-carcinogen. So the activity of your microbiota is plausibly linked to long-term risk of bowel cancer, which is why I talk about it. It's a long-term risk.
Got it.
Now, again, you could have an awful microbiota and never develop bowel cancer. It's about, we all know people who smoke 60 cigarettes a day and live to 100, right? But it's a risk, right? Your chances might increase. So, and again, it's one of these, we are not there yet as a field. I don't know what is the best microbiota to protect you from bowel cancer. So how do I know what's in your pill in 30 years' time isn't going to tip the balance and give me something dodgy? You know what I mean?
Absolutely.
But that's a personal decision. And I like to say, I suspect people who aren't happy with what they've got right now, they would happily take the pill, I'm sure.
On the bowel cancer element, I can remember, I think it was maybe 4 or 5 years ago, I walked into a meeting with you and opened a beef jerky.
Yeah.
You're like, James, what are you doing? Yes. I was like, what? This is my protein source.
Yeah.
And you were like, yeah, heavily processed, highly
meat,
processed meat.
yeah.
So is that a thing? Processed meat?
So I know we talk about nutrition science. Honestly, I get why people really get frustrated with nutrition science. In the paper, 1 week coffee will cure cancer, next week coffee gives you cancer, right? I get for the general public, I get how that is super frustrating, right? And that's science. I told you,
butter,
science,
margarine.
Yeah, exactly. Science is this long, slow process. Unfortunately, it takes a lot of time to get to the truth.
Yeah.
Processed meat is one of the few examples where, the evidence consistently suggests that, if you have a lot of it over your lifetime, your risk, doesn't mean you're definitely going to get bowel cancer, but your risk, it's like smoking, right? If you smoke, your risk of getting lung cancer increases. So no, I think, lots of processed meat is generally not great advice for most people.
Anything else people should avoid for their microbiome, general health?
Again, it's a really good question. There's lots of stuff on things like modern food ingredients and ultra-processed foods and stuff, and most of that comes from rodent model studies, and we just been doing work on emulsifiers, for example. It's unpublished, so I can't talk too much about it. But there's lots of work in rodent models suggesting emulsifiers might destroy your gut microbiota. And so the one we used, which I can't talk about, it was absolutely fine. Zero impact on the microbiota, zero impact on the blood. There was no extra transfer of bacteria. It was absolutely fine. So that emulsifier is probably absolutely fine, right? So,
That's
there's
good news.
Yeah, no, it's great news. Yeah. So so again, ultra processed foods are an easy target, right? Because they're unnatural and it's new, and new things are always, like vaccines, whatever. They're always something that people are aware of, and fair enough, right? But again, I'm an evidence-driven man. What's the evidence? Are these things definitely going to do me in?
And personally, I would avoid fructose syrups, glucose fructose syrups.
There's good reasons for that, even beyond your microbiome, right? They're full of calories. They make you gain weight. If you drink like a 2-liter bottle of 100%. Fizzy juice every day, it's 2,000 calories every day. That's your 100%. Calorific allowance. So I drink water, right?
Yeah.
Because it's wasted calories. I'd rather eat nice food and get fat that way. So I think even beyond the microbiome, there's just good reasons. And I think that's a nice thing about the microbiome advice that you can give, is it, as I said earlier, it ties into current advice quite nicely. It just adds another mechanistic layer to that.
It ties in. I take an even more optimistic view, which is that the historic health advice actually was just microbiome promoting health advice. So it's all in. Now, you published what has become, I believe, quite a popular paper.
Yeah.
With Lindsay Hall.
Yep. No, Leslie Hoyles. Sorry. Leslie.
Back up. Yeah. It's not Lindsay. It's Leslie.
Oh, Leslie Hoyles, yeah.
From Nottingham.
Yeah.
Right,
yeah,
okay.
yeah.
Back up. But you work with Lindsay, right?
Yeah, we do. Yeah,
Right, okay.
yeah, we do.
Now, you published we'll edit all that, yeah. You published a quite impactful and very popular paper with Leslie Hoyles.
Yep.
And it related to myths and misconceptions within the microbiome.
Yeah.
Could you walk us through then some of the key ones?
There's a little backstory there. So because the microbiome is super interesting, super important, it's drawn lots of attention, which has been great, as we've talked about earlier on in the chat, but it's also brought with it a lot of hype. And, I'm not big on hype. I like evidence. And so I guess, I would see statements repeatedly appearing that I knew definitely were not true. And so I guess I was asked to give a talk by a lady called Sheila Patrick, professor at Edinburgh, on myths and truths in the microbiota. So I went and I sort of skewered some of the myths that I see all the time in press, which I know are not right. And it went down really well. There was about 10 editors came after me and said, write a paper for us. So,
really?
So I wrote, the first person I asked was the nature microbiology person, so I wrote for them. And really, it's just an attempt to sort of, one, correct the record in a few things that really annoyed us. But I guess more importantly, because some of these are trivial, it's more important about just asking for slightly more credulity in microbiome research. It's very easy to get on the bandwagon and think it's going to cure everything. It's this wonder thing, and it may cure some things. But at the moment, as we talked about, there is this vacuum. It's not there yet. So I guess it's a call for a bit more critical assessment, and it's a correction of a few sort of pervasive myths, which, actually aren't true, but are very widespread in literature.
And Profiles is also a bit of a skeptic. Is that correct?
Yep.
So it's two skeptics.
Yep. I think, Leslie wouldn't argue with that.
No, I think it's true as well. I know who you're talking about.
I don't think skeptic's a hard word. I just think I'm a realistic person, but with a Scottish edge. Because I genuinely do have lots of problems for the field. I'm not. Honestly, I said before, I really hate contrarianism for the sake of contrarianism. Like people get to feel really clever about themselves by saying, "Oh, look at all those stupid scientists. What they know, they're all wrong." And usually they're not. Right. As I said, the scientific method really works, but like I say, there's a fine line between going too far based on what the evidence will tell you.
So could we clear some of them up today?
Happy to, yeah.
So what are some of the most important ones then?
So, I mean, we touched on a few already, to be honest, so I guess we can skip through them. I think the one thing that's easy to do, especially when you have 1,000 papers a month, is you don't have time to read all these papers. It's so easy just to regurgitate a fact, without actually going back and searching it. So again, if you read in the literature, Joshua Lederer invented the word microbiome. He didn't. There are 10 to the 12 bacteria per gram. There aren't. It's a new field. No, it's not. We talked about it. It started hundreds of years ago. So it's these things are just easy to repeat and parrot. People read them. They can become lodged in their brain as dogma. They get repeated. So those are the trivial ones. We got them out of the way to show that, there is a lot of mythology in microbiome science. So we skewered them. Some of the ones that are a little more important for the field are linked to the diseases that we talked about, that the microbiome must be causative in every disease. Again, it might be, but at the moment, we don't have the evidence for that. And it's been very difficult, as we talked about earlier, to disentangle cause from effect. And we need a lot more mechanistic research to really get to actual plausible mechanisms, which will lead us to treatments eventually.
With you.
Other stuff that's in there, I guess some of it's quite techie, sciencey, nerdy stuff.
Come on.
But okay, well, I'll hit you with a few. So sequencing has been great, right? It's been amazing. We talked about DNA sequencing, and it's really revolutionized the field. It has, absolutely. But it's also embedded biases and artifacts. So you read quite often, these are amazing technologies and they're perfect. No technology is perfect. Everything that you use is just a technique, no matter how powerful, and they all have limitations, caveats, drawbacks. And if you're not cognizant of those, you make some very bad conclusions and you push the field down unproductive paths. So one of it was sort of, we talk about this lack of reproducibility. When people find bug X associated with disease Y, someone repeats a study, they don't find it. The scientific method is working, but a lot of people in the meantime have jumped on that bandwagon and gone, oh, this disease must be caused by this bug, and it goes nowhere. So again, as I told you, science is difficult, it's tricky, it takes time. So a little bit of temperance sometimes on that. Things like the databases we use. So we talked about culturing and why culturing is so important. So if the cart's kind of ahead of the horse with sequencing at the moment. So we can generate all this data, but actually most of it ends up in the unknown pile. We don't have a clue what these genes do, so we ignore it. And so really what we need to do is get back in the lab and do experiments and find out what these things are doing. So it's kind of it's like a plea for a bit more of that in the field. Some things that people used to think were true. You can't culture lots of these gut bugs. Well, you can. We already have. And so things that were repeated ad nauseum actually don't have a basis in fact. So a bit more,
Is there anything else we might want to skewer? I mean, you talked about obesity earlier.
Yeah, I'm trying to think about the article now. What else do I want to skewer?
It's open access, right?
Yeah, no, it is, at the moment it is, anyone can read it, I think... read the article, I guess, is the advice. It's in there. It's quite a short read. It's only 2 or 3 pages.
It's very good. Yeah, very popular.
Thank you.
Good. Well, Alan, it's been a pleasure.
Yeah, likewise.
Thank you for having me on.
Chat, yeah.
No, thank you for coming on. Thank you, James, for having me on your podcast.
You're welcome.
No, thanks so much. Really a pleasure.
Yeah.
Really a pleasure.
No, it's love having a chat. Yeah.
Thank you so much.
It's good. Thank you.
Cheers.